Hello and welcome to SAYED'S Biology. Today I will be discussing the structure of a virus. A virus is a submicroscopic infectious agent that replicates only inside the living cells of an organism. Viruses are considered to be acellular organisms as they lack the typical features of living cells such as metabolism and the ability to reproduce on their own. They are made up of genetic material, either RNA or DNA, surrounded by a protein coat, and in some cases, an outer lipid envelope. The structure of a virus can be broadly divided into two parts, the genetic material and the protein coat. The genetic material of a virus can be either RNA or DNA, but never both. The genetic material carries all the information necessary for the virus to replicate inside a host cell. RNA viruses can have both single-stranded and double-stranded RNA molecule as their genetic material. Single-stranded RNA molecule can be either positive sense, which can be directly translated into proteins by the host cell, or negative sense, which needs to be converted into a positive sense RNA molecule before translation. RNA viruses can further be classified into three categories. They are positive sense RNA viruses, negative sense RNA viruses, and retroviruses. Positive sense RNA viruses have a single-stranded RNA molecule that can be directly translated into proteins by the host cell. Examples include the coronavirus, hepatovirus, etc. Negative sense RNA viruses have a single-stranded RNA molecule that needs to be converted into a positive sense RNA molecule before translation. Examples: avian influenza virus, rabies virus, Ebola virus, and so on. Retroviruses have a single-stranded RNA molecule that is reverse transcribed into DNA once inside a host cell. The DNA is then integrated into the host cell's genome, where it can be transcribed and translated into new virus particles. Examples include HIV and human T-cell leukemia virus. On the other hand, DNA viruses have a double-stranded DNA molecule as their genetic material. Sometimes the DNA can be found as single-stranded. Double-stranded DNA molecule can be either linear or circular. DNA viruses can further be classified into two categories. They are enveloped and non-enveloped DNA viruses. Enveloped DNA viruses have an outer lipid envelope surrounding their protein coat. Examples include herpes simplex virus and varicella zoster virus. Non-enveloped DNA viruses do not have an outer lipid envelope surrounding their protein coat. Examples include adenovirus and papillomavirus. The protein coat of a virus is also known as the capsid. The capsid is made up of individual protein subunits called capsimers. The capsid protects the genetic material of the virus and helps it enter the host cell. The shape of the capsid can vary between different types of viruses. The most common shapes are helical, spherical. The helical-shaped viruses have a helical structure capsid that forms a helix around the genetic material. Examples include tobacco mosaic virus or TMV. The spherical-shaped viruses have a capsid that forms a roughly spherical shape with 20 triangular faces. Examples include influenza virus. Some viruses have a capsid that is neither purely helical nor purely icosahedral. Examples include bacteriophage T4 and poxvirus. Some viruses have an outer lipid envelope surrounding their protein coat. The envelope is derived from the host cell's membrane and contains viral proteins that help the virus enter host cells. The envelope also protects the virus from the host's immune system. Examples of enveloped viruses include influenza virus, HIV, and herpes simplex virus. In summary, viruses are submicroscopic infectious agents that consist of genetic material surrounded by a protein coat. The genetic material can be either DNA or RNA, but not both.